So I'm Nancy Cox at the University of Chicago, and I'm going to talk a little bit about the GTEx project, which, as I mentioned several times, and Luke in particular had some really outstanding results that he was reporting, but I'm going to focus a little different way. So I'm going to give an introduction to GTEx. I'm going to talk a little bit about even the current results that we can talk about, crossing information obtained from the NHGRI catalog with information obtained from the GTEx project. But then also to go into a little bit more about how I think it will be possible in the future to use results of studies from GTEx to test additional sort of big picture hypotheses within the NHGRI catalog and in large-scale genome studies in general, both GWAS and sequencing, but studies across the entire genome. And then I'm going to just mention a few of the pitfalls that people need to keep in mind in terms of precisely those things. So in trying to test things in the NHGRI catalog and in trying to draw concrete conclusions about classes of functional variants and their enrichment in the hits that we see highlighted in the NHGRI catalog, the kinds of challenges that unquestionably arise in being able to draw conclusions from these studies. So people just need to keep in mind as we move forward. It's an outstanding resource, and our group and many others make nearly daily use of the information on the catalog, and I see it being a great resource for the scientific community in many ways going forward. So GTEx is the genotype tissue expression resource. It's really expanding now beyond just gene expression. So there was a recent RFA for additional kinds of omics technologies to be done on the tissues that are being collected through GTEx. So in GTEx, most of the tissues are being collected from organ donors, but there will be some tissues collected from surgical patients. In the sort of first round, the emphasis has been on RNA-seq studies being conducted in these tissues, but in addition to the RNA-seq, a subset of subjects and tissues, the RNA from a subset of the tissues and subjects were actually assayed for transcriptome expression on array technologies to make it more straightforward to be clear about the conversion between the array-based assessments of transcript levels and what we're picking up with RNA-seq. And similarly, the opportunity to look at some of the tissues obtained from surgical patients as compared with the same tissues obtained from organ donors will help us understand better the quality issues on the RNA from living individuals versus very recently deceased and individuals. The intent, of course, is to collect a large number of tissues on the same set of individuals. That gives us a lot of really interesting opportunities to enhance power for detecting things like eQTLs, and more generally for studying transcript biology by developmental ontologies of cells and tissues, by geographic proximity, and so forth. So there's a lot of opportunities. GTEx is well geared up now, but there are many more tissues that are shown here. So that here, this is for the latest version of the data that are publicly available. And you see, for example, we have the most samples available in whole blood, but many of the tissues have quite similar numbers of individuals available. And in general, as the sample collections increase, these tissues where we're clearly getting good quality RNA from most individuals on, this you see as we go further and further in the project, these tissues will continue to lead the way in terms of the numbers of individuals available for them. And not surprisingly, tissues like brain and pancreas, for example, that are quite fragile tissues, it's much harder to get sufficiently high-quality RNA to do the RNA-seq studies, and so there will be tissues that lag. Nevertheless, as the numbers of individuals collected increase, we are likely to get from many tissues some level of information on transcript levels. So the RNA-seq data, of course, then is supplemented by genome-wide genotyping data on, right now, the Illumina 5 million SNP chip. There's some exome sequencing also being done, and of course, in the future, it may turn out to be more cost-effective even to do sequencing in the subjects, but for now, that's the design. So the GWAS Catalog, of course, has been used by many investigators to show that classes of functional variants are enriched in SNPs associated with complex human traits. And of course, EQTLs were among the first of those to be reported as enriched among the SNPs associated with complex human traits. Methylation QTLs, which we might define as SNPs associated with methylation status at sites variably methylated. Protein QTLs, so SNPs associated with protein levels, which may or may not be independent of the messenger RNA level. So under some definitions, people are really focused more on the SNPs that are associated with protein levels independent of their association on messenger RNA levels. MicroRNA, so SNPs that are associated with microRNA levels, and we're going to see more and more information emerging on both the function of other kinds of RNA molecules, but also the regulation, so DNA variants that appear to regulate these regulatory non-coding RNAs. As John did a tour de force talk on the ENCODE annotations, I think it's safe to assume that essentially all classes of truly functional variants would be expected to be enriched among the catalog variants, with the non-coding classes of functional variants probably coming to the fore first, because the missense and nonsense variants, of course, were poorly interrogated in first-generation GWAS. It will be interesting to see how that may change as exome chips become more and more widely used in the context of GWAS. And of course, as was discussed earlier, the opportunity to start to include genome-wide interrogations on rare variants will no doubt find some way into this catalog or similar sort of catalog for characterizing classes of rare variants, missense and nonsense primarily, that might be associated perhaps sometimes at the level of the gene with complex human traits. And so this leads to, so in the sort of first results, where we can really cross the GTEx, the emerging GTEx information with information from the NHGRI catalog, one of the first obvious questions is where are the gene targets of trait-associated variants that might be regulatory? And this is one of the ways that the EQTL studies that are being done in GTEx are particularly valuable. So a lot of the annotations that have been discussed don't necessarily get you to genes. There have been a number of details in the presentations about how you can go about doing that, but of course the eQTL research is one way that you get to annotate a gene-based functionality to a variant that is implicated in disease or a related quantitative trait. And so this is one of the same slides Luke showed, and reiterates the point that John made. It is not necessarily the closest gene in physical proximity, or in the case of intronic SNPs or 3' or 5' SNPs very close to a gene, the gene that is physically closest. So again, this is a plot of the log-base 10 p-value for the eQTL. But we started out with all of the SNPs highly significantly associated with complex human traits from the catalog, expanded that list to include everything in strong LD, so R² of 0.8 or greater, to make sure that we got, we're more likely to catch causal variants. And then are just plotting the sort of, quote, best eQTL, that is the strongest eQTL among that set, and the distance from that strongest eQTL that is associated with a complex trait to its target gene. And as Luke noted, there's a substantial number that are quite distant. But the key point is really that only a small proportion are really hitting the most obvious gene that people sometimes annotate to the SNP. So there's a high proportion, in fact, that are cis eQTLs, but far more distant genes than the one that we have the most obvious physical proximity to. And of course, so as everyone has noted, this has a lot of implications for all the downstream bioinformatics studies. It's not something that's necessarily surprising to statistical geneticists. I mean, it's always been clear from the GWAS Catalog that when you have a SNP showing strong association with a complex trait, there's a whole host of SNPs that are in strong linkage disequilibrium that are either exactly as good a candidate because of essentially perfect linkage disequilibrium, or effectively as good a candidate. And to the extent that we use imputation now to interrogate essentially all variation that we know about, we're catching more, presumably, of the truly causal variants, even in these first-pass analyses. But it's often clear that the linkage disequilibrium among what we might think of as the candidate set of driver SNPs often encompasses several local genes. And so it's not at all surprising that we're seeing annotations at some distance away. But there are a few caveats to remember as well. So again, even in GTEx, where we are able to interrogate many different tissues, we're not yet interrogating even all of the tissues that we'll ultimately be able to interrogate. And even then, that will be only a subset of tissues at not nearly all stages of development and timing. And so, of course, we're going to miss some of the possibilities. So this is always just one part of the picture. But definitely an important thing to keep in mind, because one of the sort of rubs, one of the things that people have criticized about GWAS is the fact that we've gotten less biological knowledge from GWAS than you might have expected from the sheer number of discoveries that have been made. And I think a key part of that problem is that our discoveries come at the level of SNPs rather than at the level of genes. And we're still struggling to make that connection between the variants that are associated and the genes that are the driving biology for why we see the association. And I think that studies like GTEx and many of the kinds of annotations that John and Luke and others have talked about today will be the way that we make those connections. I'd like to turn now to what I think are more of the potential studies that people can be thinking about doing with what will come out of GTEx and potentially be able to be applied to the NHGRI catalog. And one of the first of these that I want to bring up is around genetic architecture. So in particular, the question is what aspects of genetic architecture should we investigate? And I am so old that I was actually at the NIH meeting where the term genetic architecture was coined, and it was a very long time ago. And back then, we were thinking about simple parts of the architecture, like was the gene for schizophrenia dominantly inherited, recessively inherited, or additively inherited. And of course, we have a completely different focus when we think about genetic architecture these days. Certainly a more sophisticated one than then, but clearly we are still blind men feeling the elephant with respect to even defining architecture. We're spending a lot more time these days thinking about the risk allele frequency spectrum, and some of this comes out of the earlier papers from the NHGRI catalog. So contrary to popular belief, it's not that the GWAS chips did not include any rare variants. So here I show the minor allele frequency spectrum of DNA variants interrogated on commonly used GWAS chips from earlier generations. So the Affymetrix 6.0 and Illumina 1M products. You see, the mode bin for minor allele frequency is actually for low frequency variants. But of course, we have little power to do very much with the low frequency variants, and it's always been appreciated that the quality of interrogation of the low frequency variants is not nearly as good as the quality of interrogation of the high frequency variants. And for all of the same reasons, of course, this carries over into the sequencing also. The fewer, when you don't know that a site is polymorphic, or the fewer examples you have of people carrying a heterozygous genotype, the harder it is to make a secure call. But of course, and then in this third panel here, the minor allele frequency spectrum of SNPs from the NHGRI catalog. This was from a while ago, but it's not going to be very different now, which is highly skewed toward the more common variants, and this is not really about testing the common disease, common variant model. This is simply an indication that we have very little power to detect associations in single variant tests for the really rare things. But we're intensely interested, of course, at this stage in human genetics and genomics in getting a better understanding of the relative contributions of rare and common variants to common diseases and their associated complex traits. What I want to talk about today, though, are new aspects of genetic architecture that I think we are characterizing through the research being done in GTEx, and it would be new ways of thinking about it. So I think that cis and trans is going to be a really interesting and key aspect of genetic architecture. Even from the early studies in GTEx, there will be trans regulatory variation, but I think there's another emergent feature that Luke mentioned briefly that I want to spend some time on today, and that's the notion of there being cross-tissue variation, so genetic variation that really cuts across tissues to regulate transcript levels for genes that are expressed in many different tissues versus single or limited tissue QTLs. And And there's some really interesting biology that I think we can investigate in the NHGRI catalog by thinking about this aspect of genetic architecture. So we can define cross-tissue eQTLs in a number of ways. So certainly SNPs associated with the same transcript across multiple tissues. SNPs associated with the same transcript in all tissues in which the transcript is sufficiently expressed. So that's an even more stringent definition. SNPs associated with the same transcript in at least a subset of the tissues in which the transcript is sufficiently expressed. So any of these would, of course, be valid definitions, and these different definitions are intended to alert you to the fact that we really do see these different kinds of patterns in real data emerging from GTEx even today. The single or limited tissue eQTLs have further nuances, so we certainly see SNPs associated with a transcript expressed in a single tissue. So there's a subset of genes that really are expressed nearly exclusively in a single tissue, sometimes even a single cell type, and of course sometimes even at a particular stage of development. And any DNA variant that is associated significantly with transcript levels in that context doesn't really have the opportunity to be observed as a cross-tissue eQTL because the transcript simply is not expressed so widely. But of course, we will also see SNPs associated with a transcript in only one or a limited subset of the tissues in which that transcript is adequately expressed. And there are clear examples of each of these types. That is, there definitely are DNA regulatory variants apparently associated with very tissue-specific transcripts. There are definitely SNPs that are clearly associated with transcript levels in one or just a few of the tissues in which that transcript really is expressed at levels that could be where you have, clear power to detect the DNA variant also as an eQTL, but it does not appear to be acting to regulate or certainly not significantly associated with transcript levels in some of the tissues where you're seeing the sufficient levels of the transcript to have been powered to pick up the eQTL effects. And so these are very interesting ways of considering investigations within the NHGRI catalog. And so I'm going to try to show you some of the data that suggests that this is actually going to be relevant for human disease as an aspect of genetic architecture. And to do that, I'm just going to show some simple investigations from within the Wellcome Trust Case Control Consortium, GWAS. So just to remind everybody, this is a very large-scale, early-stage genome-wide association study involving 7 common human diseases and a shared set of controls with about 2,000 cases for each of the 7 human diseases and a shared set of 3,000 controls. And so what I'm going to be showing you is examples of studies that we have conducted within GTEx where we're trying to understand better the relationship of these eQTLs to associations detectable in the context of GWAS. And in blue here is simply the QQ plot for the hypertension phenotype from the Wellcome Trust Case Control Consortium. And as with all of the Wellcome Trust Case Control Consortium phenotypes, except for the autoimmune ones that had clear signal at HLA, most of the rest were pretty great at defining the null here, the null for the QQ plot. So you see for the blue line, using all SNPs interrogated in the GWAS, there's really no evidence for excess signal for hypertension. In the red, what I'm showing are just cis-eQTLs identified through GTEx studies in adipose tissue. So we're using the same, so the association that's being plotted is of these SNPs and their association with the hypertension phenotype, but the only SNPs being included in the QQ plot are the cis-eQTL SNPs. And what you can see is a clear signal for adipose eQTLs in SNPs showing association with hypertension. And the excess becomes apparent fairly early in so even in the 10 to the minus 0.01 level p-value, you're seeing an excessive signal. So the similar sort of thing here I'm going to show for data from the Psychiatric Genetics Consortium. So just pulling information on attention deficit hyperactivity disorder. In blue, again, the QQ plot using all SNPs, pretty much defining the null, as we've seen for many of the GWAS. And of course, in the Psychiatric Genetics Consortium, they've aggregated a lot of the existing available GWAS, so you're really seeing results of a meta-analysis over these phenotypes, so a much larger number of individuals, still pretty much on the null. But now we're going to pull that apart and look at this with just eQTLs from different tissues available in GTEx. So we have the adipose, muscle, lung, the nerve, tibia, cerebellum, and whole blood. And one of the things I want to draw your attention to, so there's something that looks a little more different from the null here for ADHD across several of these tissues, but you'll notice that the pattern is actually so similar across, for example, adipose, muscle, and nerve, tibia, that you might be suspicious that this is driven by eQTLs that are actually cross-tissue eQTLs from many different tissues. And if you were to guess that, you would be largely correct. And in fact, you really can't draw much from the cerebellum yet. The data from brain and GTEx, it's such a small number of samples that there really isn't much power yet to even detect any QTL. But I think other groups have looked at some of this and using brain QTLs derived from other data sets, and I would suspect we'll see something similar, but perhaps even a little bit different pattern from within brain, where you would likely be picking up potentially tissue-specific QTLs as well. If we look at something, one of the traits from the MAGIC consortium, so this is HOMA-IR, so it's a measure of insulin sensitivity. Again, in a very large meta-analysis across many studies. So using all SNPs shown here in the blue, there actually is some evidence that, for there being a genetic contribution to HOMA-IR from variants, but as you have now accumulated information from many GWAS, you're really starting to see signals. But it's a much more notable signal when you consider the eQTLs from various different of the tissues from within GTEx. Adipose, muscle, lung, nerve, tibia, even a little bit here with cerebellum, and to a lesser extent, whole blood. And again, you might be suspicious that at least some of this is being driven by cross-tissue eQTLs.
A few minutes?
Yeah, just a few minutes. And so with respect to architecture related to the specificity of regulation, you might ask, with the NHGRI catalog, you might be able to ask questions like, well, multi-system disorders tend to have trait-associated SNPs that are cross-tissue eQTLs. Will disorders that seem to be more clearly about a single tissue have trait-associated SNPs that are more likely to be single-tissue eQTLs? And we're also suspicious that crossing using the NHGRI catalog signals with GTEx will allow some information on biology, perhaps novel information on biology to be observed. Keep your eye on hypertension here at the very bottom phenotype as we go through the various tissues. So in muscle, we see hypertension still pretty much on the null. Lung, still pretty much on the null. But in adipose, we pick up signal. And so the notion that the tissue where you're starting to see enrichment of signal may tell you something new about the biology of a phenotype is quite interesting. So here on the same plot you see from several different tissues. So with respect to questions relating biology to tissue, does the tissue specificity relate to developmental ontologies and shared signaling pathways? And I would note that the adipose eQTLs implicated in the hypertension GWAS yield additional support for both known biology that people appreciate for hypertension, the renin-angiotensin system, but also new biology. And so I think there's some real interesting opportunities. In GWAS, we've been criticized for picking the cherries. We all appreciate that we've got orchards of cherries, and I think using something like the catalog with the resources that are becoming available will not only allow us to make lots of cherry pies, but really set up full-scale factories for the kinds of pie making we need. And I'll just end with the pitfalls, which was not really a cherry pit pun. We need to be careful about how we adequately characterize the null distribution and enrichment studies in the catalog. Conditioning on minor allele frequency, distance to nearest gene density, appropriately counting for LD, certainly all necessary, but not necessarily adequate, because aspects of functionality are going to be shared across classes of variants, and as John reiterated, the ENCODE annotations. And so the things that we are able to do within a single GWAS don't necessarily translate so easily to the catalog, so we can certainly within a single GWAS do permutation studies that really allow us to finesse a lot of the primary pitfalls. And I think we have to be careful because the biases that can be introduced may be even more pronounced because within the GWAS catalog, we've got a much higher proportion of those things. And so the likelihood that these are going to map really disproportionately in functionally relevant parts of the genome is very clear. And with that, I'm happy to stop. People in my lab who work on this and are my faculty colleagues on these projects, but especially a shout out to the GTEx group, a really terrific group of us collaborators and colleagues.